Hello everyone, I'm FlorentGilles. I will present you my thesis project about dendrogenetic study of northern sugar maple stands, variability of growth and regeneration response to climate change. So it's conducted by Yves Bergeron from University of Quebec in Abitibi-Témiscamingue and co-directed by Julie Godbout from Forest, Wildlife and Parks Ministry of Quebec, also Igor Drobichev from UCATU. And there are contributors as Changkuo Huang from South China Botanical Garden and Fabio Gennarietti from UCAT. So about the context, we see climate changes as described by IPCC from the last decades. And there are possible population responses from it. First one could be migration, tracking our climatically suitable area result in shift in geographic range, or extinction of the population, failure to reproduce in novel environments that became climatically challenging, or if there could be adaptation, ability to persist locally in new environments through changes in genotypes. So on this project, we focus on migration possibility. So the target species is sugar maple, so as circular marshal, and we see that there is an noticeable shift in response to climate change of few species and also for sugar maple in northern limit of distribution. And there is particularity of continuous distributions in the yellow birch sugar maple in deciduous forest in south, or discontinuous distribution in yellow birch balsam fir in mixed forest. And the distribution and dominance of sugar maple make it a good model for studying response to climate change. So we'll use two big axes on this project, is dendroecology and molecular ecology. The first one, we'll use dendrochronological techniques and soil and climate data to see the variation of ring widths in time and distribution. About molecular ecology, we'll use population genetics to see population structure or demography of populations. And just to be specific, we will use formats of genes as species in ecology. So it's really a descriptive tool here. And for population structure, we'll be at inter-population scale. And for demography, we'll be in intra-population scale. And which sees a variation in genetic diversity over time and distribution. So what is the adaptative potential of sugar maple in front of this climate change? We want to see the variation in genetic diversity, radial growth. So in the north, will population at the northern edge of the range benefits from climate change or no? Because we saw reduced radial growth to the north for some species. And in the south, we can assume that it will be a decline. So what is the main objective? Is linking growth patterns, adaptation of populations, and genetic structure of sugar maple stands to better understand population dynamics, current and past, and their adaptative potential. So first, in the first big scale, we'll see pattern of growth using dendroecology tools, and also understanding current and past sugar maple branch dynamics using genetics tools with molecular ecology. And more specific scale, we will study the regeneration of northern maple populations in eastern Canada, in Quebec, using also molecular ecology. So there are three axes at different scales. So for the first chapter, it will be on climate growth relationship. We want to evaluate the growth of maples along latitudinal climatic gradients, ranging from continuous to discontinuous distribution. And we want to measure adaptation observed by dendroecology. And we hypothesize that ring widths would decrease along latitudinal transect going northward. And we want to see if the population has still adapted. The second chapter will be on genetic variability of populations. We want to establish a relationship between genetic diversity of populations and the variability of their growth response to climate. So we want to see if there is any climate growth genetic relationship. And we hypothesize that a greater genetic variability in the core, in continuous zone, than in marginal populations in discontinuous zone in the north. And greater genetic variability will result in greater variability in individual climate responses. So we want to see if there is an adaptive potential here as card in hands for the tree, and to see there are different ecotypes or not. The third part will be on genetic variability of mature individuals versus regeneration. We want to determine if the genetic diversity of regeneration decrease compared to mature trees as going northward with less reproductive trees. We hypothesize that decreased genetic diversity in regeneration due to fewer parent trees with smaller population size, and we can also check if there is any foundation effect here. And gene flow from neighboring population would be greater in the continuous zone than in the discontinuous zone. We want to see the local adaptative potential in intra-population scale. So about the study area and sampling conducted, it's conducted in Quebec, in the whole province, and it's conducted by different teams from Forest, Wildlife and Parks Ministry of Quebec. And we will focus on the yellow birch, sugar maple zone, and balsam yellow birch and balsam fir, zone. And 5 sites were added in West Quebec this summer. About Dendrogenetic data, we will do an illustration of past, present climate data with seasonal variable using some different tools as Climate Explorer, Climateana, or Geosyndics 10, sorry. And tree ring reading are already done by team from MFFP, and also ongoing work on the tree ring reading at Jupiter Lake Research Station using co-recorder and seedable softwares. We'll do some statistical analysis using generalized additive mixed model and also linear model to see growth synchronicity between trees. And about molecular ecology, we'll use genetic diversity and allele frequency to see some pattern of genetic variability. So we make some genome sampling by using single nuclear polymorphism markers, and we'll see the population structures with qualitative data, and to check the differences between population, north, south, or mature and juvenile, and also to see groups that differ with genetic diversity to check the ecotype. And also we'll study the demographic history by using quantitative data, and to check population size if there is inbreeding or drastic decrease in population size that will induce genetic diversity loss and parental contribution with mature and juvenile. So thank you for your attention.